Hello there, and welcome to Deep Sleep Seas. I'm so glad you found your way here tonight. Maybe you've had a long day, maybe you're the kind of person who prefers falling asleep to the mysteries of the ocean rather than counting sheep. Either way, you're in exactly the right place. Tonight's journey will take you somewhere few humans have ever been, down to the deepest, darkest parts of the ocean floor, where sunlight has never reached and where the pressure would crush most living things instantly. And yet in this seemingly impossible place, life not only exists but thrives in ways that challenge everything we thought we knew about what life needs to survive. Let your breathing slow. Let your body settle into comfort. Because tonight we're going to explore one of the most extraordinary discoveries in the history of marine biology. We're going down to meet the tube worms, creatures that live in total darkness, creatures that have no mouth, no stomach, no way to eat at all in the conventional sense. And yet they grow taller than a person and can live for centuries. They survive in waters laced with chemicals that would kill most other life forms instantly. They thrive in temperatures that swing from near freezing to scalding hot. They found a way to build an entire ecosystem without any connection to the sun. This is a story about resilience, about adaptation, about the stubborn persistence of life in places we never imagined it could exist. And it begins with a submarine called Alvin descending into the abyss on a February day in 1977, carrying scientists who had no idea they were about to witness something that would change biology forever. But before we descend to those depths, if you enjoy these journeys into the hidden corners of our ocean world, consider subscribing so you don't miss our next exploration. And if you're still awake enough, let me know where in the world you're listening from tonight. I read every comment. Now let's begin our descent into the deep. February 17th, 1977. The research vessel Cornell floated on the surface of the Pacific Ocean, 400 miles off the South American coast, positioned above a feature called the Galapagos Rift. Below the ship, more than 8,000 feet down, the ocean floor split along a crack in the Earth's crust where tectonic plates were slowly pulling apart. Scientists had come here searching for hydrothermal vents, underwater hot springs, that theory predicted should exist, but that no human had ever directly observed. For days leading up to this moment, the expedition had been deploying a towed camera system called ENGIS, affectionately named for its official designation as the Acoustically Navigated Geological Underwater Survey System. ENGIS was essentially a metal frame holding cameras and strobe lights, lowered on a cable and dragged slowly across the seafloor while the ship above tracked its position. The cameras clicked methodically, capturing frame after frame of the dark ocean bottom. Most of what they photographed was exactly what the scientists expected: fresh black lava, pillow basalts from recent eruptions, cracks and fissures in the volcanic rock, and the occasional sediment drift. The deep sea floor at a mid-ocean ridge was thought to be a relatively barren place, geologically active but biologically sparse. But then something appeared in the photographs that made no sense. In frame after frame, scattered across the lava, were large white shapes. At first, the scientists studying the images thought perhaps they were rocks, mineral deposits of some kind. But as they looked more closely, the shapes had texture, had form, looked almost organic. Someone suggested they might be clams. This seemed absurd. What would clams be doing out here, so deep, so far from shore, on fresh volcanic rock with no apparent food source? The images were intriguing enough that the decision was made to send Alvin down for a direct look. The submersible Alvin, a small research submarine built to withstand crushing deep sea pressure, had been prepared for descent. Inside its titanium sphere sat three people: pilot Jack Donnelly and two scientific observers, Jack Corliss and Turd Van Andel. The submarine was guided by acoustic beacons with whimsical names, Sleepy, Dopey and Bashful, positioned on the seafloor to help Alvin navigate in the pitch darkness. These transponders would ping back to Alvin's sonar, allowing the pilot to triangulate the sub's position and navigate to the specific coordinates where Angus had photographed the mysterious white shapes. As the submersible descended, the water around it grew darker, passing through layers where different creatures lived at different depths, until finally all sunlight disappeared entirely. The descent took about an hour and a half, time spent in near silence except for the hum of Alvin's life support systems and the occasional crackle of communication with the surface ship. The three men inside had only small viewports to look out of, circles of thick acrylic, giving them narrow windows into the dark world outside. As they descended, they watched the water change color, from blue to deep blue to blue-black to absolute black. Occasionally something would drift past in the lights, perhaps a jellyfish or a strange gelatinous creature, translucent and ghostly. Down here, 2.5 miles below the surface, the pressure was immense, nearly 4,000 pounds pressing on every square inch. The temperature of the seawater hovered just above freezing, cold enough that you could see your breath if the sub's interior weren't heated. This was a world of perpetual midnight, silent except for the hum of Alvin's systems, empty except for the occasional ghostly creature drifting past in the submarine's lights. The weight of the ocean pressed in on all sides of the titanium sphere, held at bay only by the strength of the metal hull, engineered and tested to withstand these incredible forces. Inside, the three men went about their work calmly, professionally, but each of them must have felt some sense of the immense gulf that separated them from the surface, from safety, from the world of light and air above. When Alvin's altimeter indicated they were approaching the bottom, Donnelly slowed the descent, bringing the submersible to hover just above the seafloor. The sub's powerful lights switched on, piercing the darkness, illuminating a landscape that looked utterly alien. Fresh black lava lay everywhere, formed into the characteristic shapes called pillow basalts, rounded lumps of volcanic rock that cooled rapidly in contact with seawater. Cracks ran through the fresh flows. Scattered sediment filled some depressions. It was a landscape shaped entirely by geology, by volcanic eruptions and tectonic forces, a place so remote and so harsh that the scientists expected to see very little life, perhaps a few sea cucumbers or brittle stars, eking out an existence on whatever scraps of organic matter drifted down from the surface waters far above. When Alvin reached the coordinates where the clams had been photographed, what the three men saw through the viewport took their breath away. Rising from cracks in the fresh black lava were plumes of shimmering water, warm water that wavered and distorted the view like heat rising from summer pavement. As minerals in the hot water met the cold ocean and began to precipitate out of solution, the water turned cloudy blue, depositing brown and yellow stains on the rocks below. The submarine's temperature sensors registered 8 degrees Celsius, 46 degrees Fahrenheit, water that was somehow warm in the ice-cold deep sea. This alone was remarkable, proof that the hydrothermal vents they'd come to find actually existed. That hot water really was flowing from the Earth's interior onto the seafloor. But it wasn't the warm water alone that astonished them. Surrounding these vents, clustered thick around the cracks from which the heated water emerged, was life, not sparse, scattered, barely surviving life clinging to existence in a harsh desert, but abundant, thriving communities of creatures the scientists had never seen before. The biomass, the sheer volume of living tissue per square meter, seemed comparable to what you might find on a healthy coral reef or in a productive tide pool. And this was staggering because there was no sunlight here, no photosynthesis, no obvious source of food to support such abundance. The large white clams from the photographs were everywhere, some as big as dinner plates, clustered so densely that their shells touched each other, creating pale carpets across the dark lava. There were mussels with deep blue shells, their bodies tucked inside, the shells hanging slightly open to filter water. There were crabs, pale yellow crabs with reduced eyes, picking their way carefully across the rocks, their legs navigating between clamshells and mussel beds. There were strange bright red anemones that looked nothing like their shallow water cousins, their tentacles waving in the gentle current created by rising warm water. There were translucent shrimp, darting about with jerky movements, their bodies catching the light from Alvin's lamps. And rising above it all, swaying gently in the current, were things that looked like enormous white tubes, some of them 8 feet tall, each topped with a brilliant scarlet plume that waved like a feathery flower in the dim glow of Alvin's lights. These structures were utterly unlike anything in the scientists' experience. Were they plants? Impossible. No plants could live without light. Were they animals? But what kind of animal looked like a white pipe with a red top? As Alvin maneuvered closer, the red plumes retracted slightly, pulling back into the protection of the tubes, a defensive response suggesting these were indeed living creatures, sensitive to the submarine's presence, capable of movement, even if they couldn't leave their tubes. These were tube worms, and though the scientists didn't know it yet as they sat there in wonder, staring at this impossible garden in the abyss, these creatures represented something revolutionary. They represented life that didn't depend on the sun. This realization would take time to fully sink in. time to understand the biochemistry and the symbiosis that made it possible. But even in that first moment of discovery, looking at these bizarre animals thriving in conditions that should have been lifeless, the scientists knew they were witnessing something profound. For as long as humans had studied biology, since the days of Aristotle and Linnaeus and Darwin, we had understood that all life on Earth ultimately relied on photosynthesis. Plants and algae captured energy from sunlight and turned it into food through the remarkable process of converting carbon dioxide and water into sugars and oxygen. Animals ate the plants, or ate other animals that ate the plants, or ate the tiny phytoplankton in the ocean that photosynthesized. But either way, every food chain, every ecosystem, every living thing on the planet could trace its energy back to the sun. The sun powered everything. This was one of the fundamental principles of ecology, taught in every biology class, accepted as universal truth. Or so we thought, but down here at the bottom of the ocean, in darkness so complete that not a single photon of sunlight had ever reached this place in the billions of years since the ocean formed, entire communities of animals were thriving. Giant white clams with flesh the color of blood, blue mussels growing densely packed in beds, towering tube worms with blood-red plumes reaching toward the black water above them, crabs and shrimp and anemones and snails, a whole ecosystem of interconnected life, and they were thriving in a toxic soup, water rich in hydrogen sulfide, a chemical that smells like rotten eggs and is poisonous to most life on Earth, a chemical that would kill a human in minutes at these concentrations. This wasn't just surprising. This wasn't just an interesting discovery of some unusual deep-sea creatures. This challenged fundamental assumptions about the nature and requirements of life itself. If these animals weren't getting their energy from the sun, either directly or indirectly through eating other organisms that got it from the sun, then where was the energy coming from? How were they surviving? What were they eating? The questions multiplied with every minute that Alvin spent hovering over this strange oasis. The scientists in Alvin that day couldn't fully explain what they were seeing. They could observe, they could document, they could marvel, but the mechanisms, the biochemistry, the symbiosis, all of that would have to wait for careful laboratory work back on land. They collected samples, using the submersible's mechanical arms to pluck specimens and place them in collection containers. They took hundreds of photographs, capturing images of the vent sites from every angle. They made careful notes of temperatures and water chemistry, measuring how conditions changed with distance from the vent openings. And all the while, part of their minds was struggling with the impossibility of what they were witnessing, life abundant and thriving, in a place where all their training said life should be scarce or absent. But the real explanation, the mechanism by which these creatures survived, would take years to fully understand. It would require bringing samples back to laboratories, culturing bacteria, analyzing tissues under electron microscopes, sequencing genes, measuring metabolic rates. It would require follow-up expeditions, years of careful research by teams of biologists, chemists, and geologists working together to piece together how these ecosystems functioned. And at the heart of that explanation, at the core of understanding how life thrived in this sunless world, were the tube worms, those crimson-tipped towers rising from the volcanic stone, creatures unlike anything in the known animal kingdom, creatures that would revolutionize our understanding of symbiosis, of metabolism, of the very requirements and possibilities of life. The discovery was compared to Columbus finding the Americas. It was called one of the most important biological discoveries of the 20th century. It would spark decades of research and hundreds of expeditions to explore and understand these hidden ecosystems at vents around the world. It would force us to expand our definitions of where and how life could exist. It would inspire new fields of research at the intersection of biology, geology, and chemistry. It would change how we thought about the history of life on Earth, suggesting that the first organisms might have originated at vents rather than in shallow sunlit pools. And it would make scientists wonder, with new urgency and new hope, if life might thrive in other dark, chemically rich environments, perhaps buried beneath the ice of distant moons, perhaps in the subsurface oceans of other worlds, perhaps even in the hot volcanic depths of a young Earth before photosynthesis had evolved. But on that February day, as Alvin ascended back toward the surface after hours on the seafloor, as the submarine climbed slowly back up through the darkness toward the light, carrying the first samples from a hydrothermal vent ecosystem, carrying specimens preserved in collection bottles and temperature-resistant containers, carrying proof that the vents existed and that they teemed with life more abundant and more bizarre than anyone had imagined possible. The three men in the submersible knew only that they had seen something extraordinary. They had no elegant theories yet, no complete explanations, no grand synthesis of geology and biology. But they had witnessed something that would change their field forever. They had glimpsed another world, a world that had been here all along, hidden in the deepest darkness thousands of feet below the surface of the ocean, existing in parallel to the sunlit ecosystems above, waiting patiently for millions of years to be found, waiting for humans to develop the technology and the curiosity to descend into the abyss and bear witness to its existence. Imagine standing on the ocean floor, if such a thing were possible, surrounded by total darkness. The pressure around you is so extreme that it would instantly crush an unprotected human body. The cold is bone deep, the water just a degree or two above freezing, and yet, if you turned on a light, you would see rising before you what look like white pillars. Some short, some as tall as a house, clustered together like organ pipes or a grove of pale, slender trees. These are the tubes that give tube worms their name, and each one is a marvel of biological engineering. The tubes are made of chitin, the same tough material that forms insect exoskeletons and lobster shells, but also other compounds, proteins and minerals secreted by the worm inside. The worm builds its tube from the inside, constantly adding new material at the top as it grows, extending its protective casing upward, year after year, ring after ring, like a tree, adding growth rings, only vertically toward the water above. The tubes can be several inches in diameter, and they're surprisingly strong, able to withstand the battering of deep-sea currents and the occasional bumping from crabs or fish. Some tubes are smooth and white, others are stained yellow and brown from minerals in the water. Still others show signs of wear, cracks and chips from a lifetime of exposure to the harsh deep-sea environment. But despite their external appearance, these tubes are not lifeless stalks. Each one contains a living animal, anchored at the bottom, its body extending up through the tube's interior. If you were to look at the top of an occupied tube, you would see the most distinctive feature of the tube worm, its plume. This structure extends from the tube's opening and spreads out like a crimson flower or like the fronds of a fern, all lacy and delicate. The plume is made up of many fine filaments, each one branched and covered with even tinier branches, creating an enormous surface area packed into a relatively small space. And the plume is intensely brilliantly red, the color of blood, because it contains a substance very similar to hemoglobin, the oxygen-carrying protein in our own blood. This plume is not a mouth. Tube worms have no mouth. The plume is an exchange organ, a specialized structure that reaches into the water and absorbs what the worm needs: oxygen from the seawater, carbon dioxide, and most importantly, hydrogen sulfide, the toxic chemical spewing from the vents. The plume does this work constantly, waving gently in the current, its thousands of branches maximizing contact with the water flowing past. Below the plume, inside the tube, the worm's body stretches downward. The body is segmented, as you might expect from an annelid worm, though you would never know it to look at one because you can't see inside the tube while the worm is alive. The body is white or pale pink, soft and vulnerable, protected by the hard tube surrounding it. And here's where things get truly strange. The worm has no mouth, as we've said, but it also has no stomach, no intestines, no digestive tract of any kind. It has no way to eat food in any conventional sense. It doesn't hunt, it doesn't graze, it doesn't filter particles from the water. So how does it survive? The answer lies in an organ that takes up much of the worm's body cavity, a spongy mass of tissue called the trophosome. This organ is unique to these worms. It doesn't exist in other annelids, not in the earthworms in your garden or the polychaete worms on coral reefs. The trophosome is absolutely packed with bacteria, billions and billions of microbial cells living inside the worm's tissue. And these bacteria are the key to everything. The largest and most famous of these tube worms is a species called Riftia pachyptala. An individual Riftia can grow to 10 feet tall, though 6 to 8 feet is more typical. They grow remarkably fast for deep-sea creatures, sometimes adding more than a yard of length in a single year when they're young, though this growth slows as they age. They can live for decades, and they do so in one of the most dynamic and dangerous environments on the planet. But Riftia is not the only tube worm. There are other species, some found at the hot volcanic hydrothermal vents, others in cooler environments called cold seeps where chemicals leak more slowly from the seafloor. These different species have adapted to their particular environments, growing at different rates, living to different ages, favoring different chemistries. But they all share the same basic body plan: the protective tube, the crimson plume, the trophosome packed with bacterial partners, and the complete absence of a digestive system. When you see photographs or video of tube worm colonies, what strikes you is how densely they grow, how close together the tubes stand. In productive vent sites, hundreds or thousands of tubes may cluster in a relatively small area, each worm competing for space and for access to the vent fluids. The worms can remodel their tubes, both at the top and at the base, which gives them some ability to adjust as they grow and as neighbors crowd in around them. Some tubes lean, some stand straight, some are taller, some shorter. Together they create a kind of underwater forest, swaying gently in the current, a forest of crimson-topped white towers growing from cracks in the volcanic rock. These worms have no eyes. For what good would eyes do in perpetual darkness? They have no need to see, no brain complex enough to process images, even if they had the organs to capture light. They are, in a sense, simple creatures, at least in terms of their nervous system and sensory apparatus. But in other ways, in the ways that matter for survival in their extreme environment, they are sophisticated beyond measure. They have solved problems that would kill almost any other animal. They have forged a partnership with bacteria that allows them to thrive where nothing else can. They have become, against all odds, some of the most successful creatures in the deep sea. And they have done this without ever tasting a single bite of food, without ever opening a mouth that doesn't exist to consume another organism. Instead, they have become living vessels for bacterial gardens, creatures that grow inside them and that feed them from within, converting the toxic chemicals of the vent fluids into the nutrients the worm needs to build its body, maintain its tissues, grow its tube, and extend its plume into the dark water above. To understand why tube worms exist at all, we need to understand the place where they live, the hydrothermal vents themselves. These are not gentle springs or calm seeps. They are violent, dynamic features, where the Earth's interior heat breaks through to the ocean floor, where chemistry happens at extremes, where the very rock beneath is young and active and constantly changing. Hydrothermal vents form along mid-ocean ridges, the vast underwater mountain ranges that snake across the floor of every ocean basin, totaling more than 40,000 miles of interconnected volcanic ranges. These ridges mark the places where tectonic plates are pulling apart, where magma from the Earth's mantle rises up to fill the gap, cooling as it reaches the water to form new oceanic crust. This is the engine of plate tectonics, the fundamental process that has been reshaping the Earth's surface for billions of years, moving continents, building mountains, creating ocean basins, and it's happening right now, continuously, in the depths of the ocean where most people will never see it. The process works like this as the tectonic plates pull apart, driven by convection currents in the Earth's mantle. They create a gap at the spreading center. Magma, molten rock from deep within the Earth, rises to fill this gap. When the magma reaches the cold seawater, it cools rapidly, solidifying into basalt, the dark volcanic rock that forms the ocean floor. But the cooling happens from the outside in, creating characteristic shapes. When lava erupts underwater and cools quickly in contact with seawater, it forms rounded structures called pillow basalts, looking like a pile of giant pillows stacked on top of each other. When lava flows more continuously, it can create sheet flows, smooth expanses of volcanic rock. And when lava erupts violently, it can shatter into fragments that create breccias and other volcanic deposits. As the tectonic plates pull apart, cracks form in the newly created rock. These cracks are pathways, routes through which seawater can penetrate down into the crust. And this is where the magic begins, where geology creates the conditions for the vent ecosystems. Seawater seeps down through these cracks, descending deep into the crust, sometimes penetrating a mile or more below the seafloor. The journey downward is slow, the water filtering through porous rock, through fractures, through tiny spaces in the newly formed crust. And as it descends, the water encounters extreme heat from the magma chambers below, from the hot rock that's just solidified, from the geothermal gradient that increases with depth. The water can reach temperatures of 750 degrees Fahrenheit, sometimes even hotter, temperatures at which water would normally boil instantly. But at these depths, with the immense pressure of the overlying water and rock pressing down from above, something remarkable happens. The superheated water doesn't boil. Pressure keeps it liquid, even at temperatures far above water's normal boiling point. Under these extreme conditions of heat and pressure, the water's properties change. It becomes a powerful solvent, far more aggressive than normal seawater in its ability to dissolve minerals. The water strips metals from the rock through which it flows: iron, copper, zinc, manganese, lead, silver, gold, and many others. It picks up sulfur from sulfide minerals in the basalt, combining with it to form hydrogen sulfide. It leaches silica from the volcanic glass. It becomes acidic as it heats, and as it does, it becomes even more effective at dissolving minerals. The chemical composition of the water is transformed completely. What started as relatively normal seawater, with its familiar mix of sodium and chloride and other ions, becomes something entirely different. A hot acidic solution loaded with dissolved minerals and gases, a chemical-rich brew that carries with it a sample of the Earth's interior chemistry. Then, buoyant because it's hot, and therefore less dense than the surrounding water, this hydrothermal fluid begins to rise back up through the crust. It flows upward through cracks and channels, driven by convection, seeking a path to the ocean floor. And when it finds one, it erupts from the seafloor as a hydrothermal vent. The most dramatic of these vents are called black smokers, and they earn their name through a spectacular process. As the superheated fluid, sometimes reaching temperatures of 650 degrees Fahrenheit or even higher, bursts from the seafloor and meets the ice-cold seawater, hovering around 37 degrees, the temperature differential creates immediate and dramatic changes. The dissolved minerals that were stable in the hot, pressurized environment below suddenly find themselves in cold water at lower pressure, and they can no longer remain dissolved. They precipitate out of solution instantly, forming tiny particles that cloud the water, making it look like black smoke pouring from the vent opening. These particles are primarily metal sulfides, compounds of iron, copper, zinc, and other metals combined with sulfur, and they create a billowing plume that rises from the vent like smoke from a chimney. These particles don't just drift away into the ocean. Many of them settle on the rocks around the vent opening, accumulating layer after layer, building up over time. As more and more mineral-rich fluid flows through the same opening, more and more minerals precipitate and settle, and gradually these deposits form structures, chimney-like formations that grow taller year by year. Some black smoker chimneys can reach several stories tall, towering structures of deposited sulfide minerals through which superheated water continues to flow. The chimneys are made of the same materials that we mine from ancient vent deposits on land when we extract copper and other metals, rich concentrations of valuable minerals created by the hydrothermal process. The chemistry happening at these black smokers is intense and complex, a dynamic mixing zone where two very different kinds of water meet and react. Hydrogen sulfide is abundant in the vent fluids, reaching concentrations that would be instantly lethal to most marine life. So is methane, bubbling from the depths. There are ammonia and hydrogen gas, both reduced compounds with energy-rich electrons ready to be transferred. There are metals in various oxidation states, iron as ferrous iron, manganese, copper, all in forms that can participate in chemical reactions. The pH can range from acidic near the vent opening, where the hot fluids emerge, to normal seawater pH just a short distance away where mixing is complete. And the temperature gradient is extreme and abrupt, from hundreds of degrees at the vent opening to near freezing just a few feet away, creating a narrow zone of intermediate temperatures where life can gain a foothold. But not all vents are black smokers. There are also white smokers, where cooler fluids precipitate different minerals that create white or pale gray plumes. There are diffuse flow vents, where the hot water mixes with seawater underground before seeping more gently from the seafloor, creating large areas of warm rock and sediment rather than focused jets of scalding fluid. And there are countless small cracks and fissures from which warm, chemically altered water flows, creating gradients of temperature and chemistry across the vent field. These different types of vents create different microhabitats. Some areas are too hot for most life. The water at the very mouth of a black smoker can reach temperatures that denature proteins and break down cell membranes. No complex life can survive in direct contact with fluid that hot. But just inches away, where the vent fluid has mixed enough with seawater to cool down, conditions become tolerable and life moves in. The tube worms favor these zones of mixing, the places where vent fluid and seawater combine to create water that's warm but not scalding, water that's rich in chemicals but not overwhelmingly toxic. They anchor themselves in cracks in the fresh volcanic rock, their roots gripping the basalt, their tubes extending upward into the zone where both warm vent fluid and cold oxygenated seawater are accessible. This positioning is crucial. The worms need the hydrogen sulfide from the vents to feed their bacterial symbionts, but they also need oxygen from the seawater, because the bacteria use oxygen in their metabolism. Finding the right spot where both resources are available is essential to the worm's survival. Hydrothermal vents are not permanent features. The volcanic activity that creates them is episodic and localized. A vent field might be active for a few decades, or in rare cases for a century or more, but eventually the flow of magma shifts, the cracks seal, the water flow slows and stops. When this happens, the vent field dies. The water cools. The chemicals stop flowing, and without their energy source, the chemosynthetic communities collapse. The worms die. The bacteria starve. The crabs and fish move on or perish. Within months or years, a once thriving vent field can become cold and lifeless, a graveyard of empty worm tubes and mussel shells scattered across barren rock. But somewhere else, perhaps hundreds of miles away, a new vent begins to flow. Fresh lava erupts onto the seafloor. New cracks open. Hot water begins to seep upward. And gradually, somehow, despite the vast distances and the seeming impossibility of it, larvae from dying vent fields find the new vents and settle there. Tube worms begin to grow. Bacteria colonize the rocks. Mussels and clams arrive. And within a few years, a new oasis of life appears in the abyss. The discovery of hydrothermal vents and their ecosystems didn't just surprise scientists, it challenged assumptions. Before 1977, most researchers believed that the deep sea was largely barren, a cold, dark, nutrient-poor desert, where life eked out a meager existence on the scraps that drifted down from the sunlit surface waters far above. The discovery that entire productive ecosystems could exist on the seafloor, ecosystems rivaling the biomass and biodiversity of some surface environments, was revolutionary. It meant the deep sea was far more complex and interesting than anyone had imagined. It meant that life was more resilient and adaptable than we'd given it credit for. And it meant that our understanding of biology, of ecosystems, of the very requirements for life, needed to be expanded and revised. All life needs energy. This is one of the fundamental truths of biology. To build and repair cells, to grow, to move, to reproduce, organisms need a source of power to drive the chemical reactions of metabolism. For most life on Earth, that energy ultimately comes from the sun. Plants and algae capture sunlight through photosynthesis, using the energy in photons to split water molecules and fix carbon dioxide into sugars. These sugars are food, stored chemical energy that can be broken down later to power cellular processes. Animals eat the plants, or eat other animals that eat plants, but either way, the energy flows from sunlight through photosynthesizers to the rest of the ecosystem. Photosynthesis is so central to life as we know it that for a long time, scientists assumed it was essentially universal. Yes, there were a few bacteria that could use chemical energy, but these seemed like minor exceptions, marginal organisms eking out livings in unusual places. The real action, the main show, was photosynthesis and the ecosystems it supported. But at hydrothermal vents, there is no sunlight, has never been sunlight, will never be sunlight. The depth is too great, the water above too opaque. Not a single photon penetrates to these depths, and yet the tube worms grow. The bacteria thrive. How? The answer is chemosynthesis, a process fundamentally different from photosynthesis, but serving the same ultimate purpose, capturing energy and using it to build organic molecules that can fuel life. Instead of using light energy to drive reactions, chemosynthetic organisms use chemical energy, specifically the energy released when reduced chemicals react with oxidized chemicals. Let's break this down, because it's both simpler and more remarkable than it might sound. The vent fluids are full of reduced chemicals, chemicals that have electrons they're ready to give away. Hydrogen sulfide is a prime example, so is hydrogen gas, so is methane. These molecules are energy-rich in a chemical sense because they can be oxidized. The seawater, meanwhile, contains oxygen, which is eager to accept electrons to be reduced. When you bring reduced chemicals and oxygen together, they react spontaneously, releasing energy as they do. This is, at its heart, similar to burning. When you burn wood or gasoline, you're combining carbon-containing molecules with oxygen, and the energy released in that reaction produces heat and light. Chemosynthetic bacteria do something similar, but more controlled and more elegant. They take the hydrogen sulfide from the vent fluids and the oxygen from the seawater, and they use specialized enzymes to manage the reaction between them. Instead of producing heat and light, they capture the released energy in molecular form, typically as ATP, the universal energy currency of cells, and then they use that captured energy to drive other reactions, including the most important one, the fixation of carbon dioxide into organic carbon. The bacteria take CO2, an inorganic molecule, and combine it with the energy they've harvested from oxidizing hydrogen sulfide, and they build sugars and other organic molecules. These molecules are food. They're the building blocks and fuel that the bacteria need to grow and reproduce, and crucially, they're also food for the tube worms. The equation for this process, simplified, looks like this: hydrogen sulfide plus carbon dioxide plus oxygen yields organic matter plus sulfur plus water. It's chemically different from photosynthesis, but functionally similar. Both processes capture energy. Carbon dioxide gets fixed into organic molecules, and life is powered. There are other chemosynthetic pathways as well. Some bacteria oxidize methane. Some use iron or manganese. Some use ammonia or nitrite. The beauty of chemosynthesis is that it's flexible. Different bacteria have evolved to exploit different chemical reactions, to harvest energy from different sources. Wherever there's a chemical disequilibrium, wherever reduced and oxidized chemicals can be brought together, there's potential for chemosynthetic life. But here's the key thing about tube worms and their bacterial partners. The bacteria don't just live near the worms or on their surfaces, though some bacteria do inhabit those spaces as well. The crucial bacteria, the ones that actually feed the worm, live inside its body, packed densely into the trophosome. This relationship is called endosymbiosis, and it's one of the most intimate partnerships in nature. The bacteria live their entire lives inside the worm's tissue. The worm provides them with a safe, stable environment, protected from predators and from the harsh variability of the vent environment. The worm also provides the bacteria with everything they need for chemosynthesis. The plume absorbs hydrogen sulfide, oxygen, and carbon dioxide from the water. Special proteins in the worm's blood, including a unique form of hemoglobin, bind these molecules and transport them through the worm's circulatory system to the trophosome. The trophosome is an extraordinary organ. It's large, taking up much of the worm's body cavity, and it's densely packed with bacterial cells. These bacteria are so abundant that they can make up more than half the worm's body weight. They live inside specialized cells called bacteriocytes, which provide each bacterium with a supportive environment. And there, inside the worm, the bacteria carry out chemosynthesis using the hydrogen sulfide, oxygen, and carbon dioxide delivered by the worm's circulatory system to produce organic molecules. But here's the elegant part. The bacteria produce more organic molecules than they need for their own metabolism. They're essentially overproducing food, and that excess food leaks out of the bacteria, or in some cases the worm digests some of the bacteria, and either way the worm absorbs the nutrients. The worm doesn't have a mouth or digestive system, because it doesn't need them. It has a bacterial garden inside its body, and it harvests the products of that garden directly. This is why tube worms can grow so large and so fast. They're not limited by how much food they can find or catch or filter from the water. As long as the vent fluids keep flowing, as long as hydrogen sulfide and oxygen are available, the bacteria keep producing food, and the worm keeps growing. A young Riftia pachyptera, living at a productive hydrothermal vent, can grow 33 inches in a single year, making it one of the fastest growing marine invertebrates known. This is possible only because of the productivity of its bacterial symbionts converting chemicals into biomass at a remarkable rate. The discovery of this system of chemosynthetic bacteria living in symbiosis with animals was as revolutionary as the discovery of the vents themselves. It showed that evolution had found a completely different solution to the problem of getting energy, a solution that didn't require sunlight, that could work in total darkness, that could turn toxic chemicals into food. It expanded our understanding of what's possible in biology, and it made scientists wonder how many other forms of symbiosis, how many other unexpected partnerships, might be hiding in the world's ecosystems, waiting to be found. The bacteria in tube worms are sulfur-oxidizing bacteria, and they're related to free-living bacteria that can be found in other sulfur-rich environments. But at some point in evolutionary history, these bacteria and the ancestors of tube worms entered into partnership. The bacteria gave up their free-living lifestyle, and the worms gave up their ability to feed conventionally. Each became dependent on the other, each specialized for their role in the partnership. And together they created something neither could achieve alone, the ability to thrive in one of the most extreme and inhospitable environments on the planet. The relationship between tube worms and their bacterial symbiotes is so complete, so essential to both organisms, that neither can survive without the other. An adult tube worm deprived of its bacteria would starve, having no way to obtain nutrients. The bacteria, meanwhile, have become so specialized for life inside the worm that they can no longer survive on their own in the environment. This is obligate symbiosis in its purest form, two organisms so intertwined that they function almost as a single unit. But this raises fascinating questions. How do the worms acquire their bacteria? Are the bacteria passed from parent to offspring, inherited like mitochondria? Or does each generation of worms have to find and capture its bacterial partners anew? The answer, it turns out, is the latter, and it's remarkable. Tube worm larvae are born without bacteria. They hatch as tiny, free-swimming creatures, drifting in the deep sea currents, and at this stage they have no symbionts, no trophosome, no way to survive for very long. They're living on stored energy from their egg, and they need to find a vent, settle down, and acquire bacteria quickly before they run out of resources. When a larva finds a suitable spot near a vent, it settles and begins to transform. Its body changes, developing the structures it will need as an adult. And somehow, during this transformation, it acquires its bacterial partners. The bacteria enter the larva's body from the surrounding environment. Scientists have found free-living bacteria around vents, bacteria genetically similar to the symbionts, and the working hypothesis is that the settling larvae somehow take in these free-living bacteria, perhaps through temporary openings in their body wall, perhaps through tissues that later close off. Once inside, the bacteria take up residence in developing tissue that will become the trophosome. They multiply rapidly, filling the organ, establishing the population that will feed the worm for the rest of its life. This is horizontal transmission of symbionts, passed from environment to host, rather than vertical transmission from parent to offspring. It seems risky, requiring larvae to find both a suitable vent site and the right bacteria, but apparently it works well enough, as tube worms are abundant at productive vents. The worm's hemoglobin is particularly fascinating. Hemoglobin, the protein that makes blood red, normally carries oxygen. In humans, hemoglobin picks up oxygen in the lungs and delivers it to tissues throughout the body. But tube worm hemoglobin is special. It can carry not only oxygen, but also hydrogen sulfide, despite the fact that these two substances normally react with each other. Hydrogen sulfide tends to bind to the oxygen binding sites in hemoglobin, poisoning the protein and preventing it from carrying oxygen. This is part of why hydrogen sulfide is toxic. Tube worm hemoglobin has evolved to avoid this problem. It has separate binding sites for oxygen and for hydrogen sulfide, keeping them apart until they reach the trophosome. It's a molecular marvel, a protein that can juggle two reactive chemicals, keeping them from interacting until the moment they're needed. When the blood reaches the trophosome, the hemoglobin releases both oxygen and hydrogen sulfide to the bacteria, which then use them in chemosynthesis. The worm also has to deal with sulfur, the byproduct of chemosynthesis. As the bacteria oxidize hydrogen sulfide, they produce elemental sulfur and sulfate. Some of this sulfur is released back into the water, expelled through the plume, but some of it accumulates in the trophosome as tiny crystals of pure sulfur. These crystals can build up over time, especially in older worms, creating a kind of sulfur storage system. Scientists believe this stored sulfur might serve as a buffer, a reserve that the bacteria can use during periods when hydrogen sulfide is less available from the vents. The partnership requires constant management. The worm has to regulate the supply of chemicals to its bacteria, making sure they get enough hydrogen sulfide and oxygen to keep producing food. but not so much that toxic levels build up. The bacteria, meanwhile, have to produce enough excess food to support the worm's needs without compromising their own survival. It's a delicate balance, maintained by chemical signals and physiological controls that scientists are still working to fully understand. What's clear is that this symbiosis is ancient. In evolutionary terms, genetic studies suggest that tube worms and their bacterial partners have been associated for millions of years, long enough for both organisms to adapt extensively to the relationship. The worms have lost their digestive system because they don't need it anymore. They've developed the trophosome, an organ that exists nowhere else in the animal kingdom. They've evolved specialized hemoglobin and other proteins to manage the chemistry of the partnership. The bacteria, meanwhile, have become so specialized for life inside the trophosome that they've lost the ability to survive outside it. This is coevolution at its most intimate, two lineages shaping each other over deep time, becoming increasingly specialized and increasingly interdependent until the line between where one organism ends and the other begins becomes blurry. The tube worm and its bacteria are partners. Yes, but they're also, in a functional sense, parts of a single metabolic system, each providing what the other lacks, together achieving what neither could alone. And this partnership has been spectacularly successful. Tube worms are among the most abundant and successful animals at hydrothermal vents. They can grow to huge sizes, they can build up dense populations, and they provide structure to the vent community, their tubes creating habitat that other animals use. All of this is made possible by bacteria smaller than the width of a human hair, living invisibly inside the worm's tissues, quietly converting toxic chemicals into the nutrients that fuel one of the deep ocean's most remarkable forms of life. Not all tube worms live at hot, turbulent hydrothermal vents. Some have found a different kind of environment, a slower, quieter, more stable ecosystem built on the same basic chemistry but manifesting in an entirely different way. These are the cold seeps, and the worms that live there have achieved something even more remarkable than their hydrothermal cousins. They've achieved longevity so extreme that it rivals the longest-lived organisms on Earth. A cold seep is not cold in the sense that its water is colder than the surrounding ocean. In fact, the water temperature at seeps is usually about the same as the ambient deep sea temperature, hovering just a few degrees above freezing. The cold in cold seep is relative, distinguishing them from the scalding hot hydrothermal vents, where hydrothermal vents are dynamic and volcanic. Cold seeps are gentle and sedimentary. Cold seeps form in different geological settings than hydrothermal vents. They typically occur along continental margins where tectonic activity or sediment compression squeezes hydrocarbon-rich fluids from deep deposits up through the seafloor. These fluids can contain methane, hydrogen sulfide, and other chemicals that originated as organic matter, the remains of ancient marine organisms that died, sank, and were buried in seafloor sediments millions of years ago. Over time, heat and pressure transformed this organic matter into hydrocarbons, and now, where geological forces permit, these hydrocarbons seep slowly back into the ocean. The flow at cold seeps is much gentler than at vents. There's no violent plume, no black smoke, no dramatic chimneys. Instead, methane and other gases bubble slowly from the sediments, or they dissolve in pore water that percolates up through the mud. The seepage can continue for centuries or even millennia, far longer than the typical lifespan of a hydrothermal vent. This stability makes cold seeps very different places to live. The tube worms of cold seeps belong to different species than those at hydrothermal vents. The most studied is Lamellibrachia loimesi, found in the Gulf of Mexico at depths of around 1,800 feet. These worms look similar to their hydrothermal cousins, with white tubes and red plumes, but they grow much more slowly and live far longer. How much longer? The answer astonished researchers when they first measured it. An average Lamellibrachia loimesi that's 2 meters long, about 6.5 feet, is estimated to be between 170 and 250 years old. Some individuals, the largest ones, may be 300 years old or more. This makes them among the longest-lived animals on Earth. Think about that for a moment. A tube worm living today in a cold seep in the Gulf of Mexico might have settled there in the early 1800s, in the time of Napoleon, when Beethoven was composing symphonies and Thomas Jefferson was president. That worm has been standing in the same spot, its tube anchored in the same patch of seafloor, for longer than the United States has been a country. It has filtered water through its plume for centuries. Its bacterial symbionts have been chemosynthesizing for generations upon generations, and it's still alive, still growing, still adding millimeters to its tube each year. Another cold seep species, Escarpia laminata, lives even deeper in the Gulf of Mexico, between 3,000 and 10,000 feet down. Studies suggest these worms regularly live 100 to 200 years, with some individuals possibly exceeding 300 years, and the largest specimens potentially reaching 400 years or more, though this upper limit remains uncertain. Why do these worms live so long? The answer seems related to their environment and life history. Cold seeps are stable. The chemical supply is steady and predictable. There's no risk of the vent suddenly shutting off or being buried by a lava flow. Predation is rare at these depths. Disease seems uncommon. Once a tube worm settles at a cold seep, finds the right spot, acquires its bacteria, and begins growing, it faces very few external threats. Its mortality rate, the chance it will die in any given year, is remarkably low, less than 1% in some populations. In environments where death from external causes is rare, evolution favors longevity. Natural selection acts on organisms that can reproduce over longer time spans, that can keep producing offspring year after year for decades or centuries. In such environments, there's no advantage to aging quickly or dying young. Better to invest in maintenance, in keeping your cells and tissues functioning well, in repairing damage as it occurs, and continuing to reproduce for as long as possible. This is exactly what cold seep tube worms appear to have done. They grow slowly, adding just a few centimeters to their tubes each year. They mature late, taking many years before they're old enough to reproduce. But once they start reproducing, they can continue for centuries, releasing eggs and sperm into the water year after year, contributing to the genetic diversity and population size of their species. The contrast with hydrothermal vent worms is stark. Riftia pucatilla, at hot vents, grows fast and lives relatively short lives, perhaps just a few decades at most. The environment demands this strategy. Vents are ephemeral, lasting perhaps 10 or 20, or in rare cases 50 years before they die. A vent worm needs to grow quickly, reach reproductive maturity fast, and produce as many offspring as possible before the vent shuts off and the colony dies. Speed is essential. Longevity is irrelevant if your habitat disappears before you could use it. But at cold seeps, where the habitat is stable for thousands of years, the optimal strategy is different. Slow, steady growth, late maturity, long reproductive lifespan, low mortality. This strategy requires physiological adaptations that slow aging, that maintain cellular function over centuries, that prevent or repair the accumulated damage that kills most organisms. Scientists are intensely interested in understanding how cold seep tube worms achieve this. What cellular mechanisms do they use? What genes are involved? Could these mechanisms teach us something about aging in other organisms, perhaps even in humans? The study of these worms' longevity required innovative research methods. You can't just keep a tube worm in an aquarium for 200 years to see how long it lives. Instead, researchers went to the Gulf of Mexico in submersibles, found active cold seeps, and carefully marked individual tube worms by staining their tubes with a bright blue dye. Then they returned a year later and collected some of the marked worms, measuring how much new, unstained tube had grown. By comparing the amount of growth in a year with the worm's total length, they could estimate its age. The results were consistent across different sites and different individuals. The worms grew slowly, just a few centimeters per year, and the rate slowed as they aged. By working backward from growth rates, researchers could estimate that a 2-meter worm was around 200 years old, and larger worms were even older. The estimates were supported by population models that included birth rates and death rates, and everything pointed to the same conclusion: these worms are extraordinarily long-lived. Cold seeps support not just tube worms but entire communities of chemosynthetic life. There are mussels of the genus Bathymodiolus, which also house bacterial symbionts in their gills and can grow quite large at seeps. There are clams, crabs, shrimp, and various other invertebrates, many of them specialists found nowhere else. There are also microbes, free-living bacteria and archaea in the sediments that participate in the cycling of methane and sulfur, contributing to the complex biogeochemistry of the seep environment. The methane seeping from the seafloor is a potent greenhouse gas, but most of it never reaches the atmosphere. The bacteria in the sediments consume it. oxidizing methane, and in the process preventing its escape to the water column and eventually to the air. This microbial methane consumption is a significant part of the global carbon cycle, a hidden process that shapes our planet's chemistry and climate. But it's the tube worms, standing like sentinels in the cold and dark, that capture the imagination. They're proof that life doesn't have to be fast or frantic to be successful. In the right environment, slow and steady wins, and they're reminders that the deep sea, which once seemed like a barren wasteland, is actually full of life living at timescales that dwarf human experience. Creatures that measure their lives not in years or decades, but in centuries, quietly thriving in the eternal darkness of the abyss. Tube worms don't live alone. While they may be the most conspicuous residents of vent and seep communities, they're part of complex ecosystems that include hundreds of other species, many of them just as specialized and remarkable as the worms themselves. At hydrothermal vents, one of the first things you notice beyond the tube worms are the mussels and clams. These bivalves cluster around vents, sometimes forming dense beds that cover square meters of seafloor. Like the tube worms, many of these mussels and clams harbor chemosynthetic bacterial symbionts, usually in their gills. The bacteria live in specialized gill tissue, and the bivalve provides them with access to both oxygen from the water column and hydrogen sulfide or methane from the vent fluids. In return, the bacteria provide the bivalve with nutrition. The giant white clam, Calyptogena magnifica, was one of the first vent animals discovered in 1977. These clams can grow quite large, up to a foot long, and their flesh is blood red because it contains hemoglobin similar to that in tube worms. The hemoglobin helps transport both oxygen and hydrogen sulfide to the bacterial symbionts in the gills. The clams extend their fleshy foot down into cracks in the rock, positioning themselves where they can access vent fluids while their shells remain protected in the cooler water above. At cold seeps, Bathymodiolus mussels are among the dominant animals. These mussels can contain not just one, but multiple species of bacterial symbionts, some that oxidize methane and others that oxidize hydrogen sulfide, giving the mussels metabolic flexibility to exploit whatever chemicals are available. The mussels attach to rocks or even to tube worms' tubes using strong threads, creating hanging gardens of blue-black shells swaying in the current. Then there are the crabs. At many Pacific vents, a pale yellow crab called Bythogrea thermidron scuttles across the lava flows, scavenging whatever organic matter it can find. These crabs are omnivores, eating bits of tube worm plume, preying on smaller animals, scavenging dead creatures that fall from the water above. They're blind, or nearly so, with reduced eyes that can only detect light and dark, which is all that's useful in the permanent night of the deep sea. At other vent sites, particularly in the Western Pacific and Indian Oceans, there are yeti crabs, so called because their claws and legs are covered with dense mats of hair like setae. These setae are colonized by chemosynthetic bacteria, and the crabs appear to cultivate and harvest these bacterial gardens, using specialized mouthparts to scrape bacteria from their setae and eat them. The crabs wave their claws in the vent fluids, apparently farming the bacteria, providing them with chemicals, and then consuming them for nutrition. Perhaps the most extreme animal at vents is the Pompeii worm, Alvinella pompeiana. This polychaety worm builds tubes on the sides of black smoker chimneys, living in water that can reach 175 degrees Fahrenheit, close to the upper limit for complex life. The worms are covered in bacteria on their backs, forming a fuzzy coat, and these bacteria may provide some insulation or detoxification, helping the worms survive the intense heat and chemical toxicity. Pompeii worms have proteins in their cells that remain stable at high temperatures, similar to proteins found in thermophilic bacteria, and they represent some of the most heat-tolerant animals on Earth. There are also vent shrimp, which can swarm in huge numbers at some vent sites, particularly in the Atlantic. These shrimp lack functional eyes, but they have photoreceptors on their backs that can detect the faint glow of infrared radiation emitted by the hot vent fluids. This allows them to avoid getting too close to the deadly heat while still staying near enough to harvest bacteria. The vent and seep communities include fish, though not many species. There are vent fish like Thermarsis cerberus, a small eel-like fish that hunts invertebrates among the rocks. There are various kinds of zorsids and bythids, fish families that have colonized vents and seeps and adapted to their particular conditions. These fish are predators, feeding on the abundant invertebrates, and they represent some of the top consumers in these ecosystems. And there are countless smaller creatures, copepods, tiny crustaceans that graze on bacteria, polychaete worms that burrow through bacterial mats, snails that rasp bacteria from rock surfaces, amphipods, isopods, limpets, anemones, and many others, each filling a niche, each part of the community's complex food web. What's remarkable about these communities is their productivity. Despite being in the deep sea, despite the lack of sunlight, vent and seep ecosystems can support biomasses comparable to productive shallow water environments. This is possible because chemosynthesis at vents and seeps is so productive. The bacteria are converting chemical energy into organic matter at high rates, and this organic matter flows through the food web, supporting dense populations of animals. The tube worms themselves play an important role in the community structure. Their tubes provide hard surfaces in an environment dominated by soft sediment or fresh lava. Other animals attached to the tubes hide among them, use them as shelter. The worms' plumes, extending into the water, create flow patterns that affect how chemicals and water move through the community. And when worms die, their tubes remain, at least for a while, providing continued habitat until they eventually erode away. The food webs at vents and seeps are unlike those almost anywhere else on Earth. At the base are the chemosynthetic bacteria, both the symbionts inside animals and the free-living bacteria in the water and on rock surfaces. These bacteria are the primary producers, equivalent to plants in terrestrial ecosystems. Herbivores graze on the bacteria or house them as symbionts. Predators hunt the herbivores, scavengers clean up the dead, nutrients cycle, energy flows, and all of it happens without any input from sunlight, driven entirely by the chemistry of the Earth's interior. But the communities aren't entirely isolated from the rest of the ocean. Organic matter drifts down from the surface waters, dead plankton and other particles that have sunk from the sunlit zone far above. Some vent and seep animals feed on this material as well as on locally produced food. So the ecosystems are, to some degree, subsidized by photosynthesis happening hundreds or thousands of feet above them. But the core of the ecosystem, the foundation on which everything else is built, is chemosynthesis, and that makes these communities unique. Reproduction in the deep sea presents unique challenges, and tube worms have evolved fascinating strategies to meet them. These are animals that cannot move once they settle. They spend their entire adult lives anchored to one spot on the seafloor, unable to hunt for mates, unable to travel to new locations. So how do they reproduce, and how do their offspring find new vents to colonize? Adult tube worms are either male or female, though telling them apart externally is impossible. Their reproductive organs develop internally, testes or ovaries depending on the individual's sex, and these produce sperm or eggs that mature over time. When conditions are right, probably triggered by environmental cues that scientists are still working to identify, the worms release their gametes into the water. Males release sperm in large clouds. In some species, the sperm cells clump together into structures called spermatosomata, packets of sperm that swim through the water as coordinated units, moved by the collective beating of all the individual sperm tails. These packets might help the sperm survive longer in the harsh vent environment, though they break apart fairly quickly. Females release eggs, typically keeping them in their tubes initially. Fertilization can happen inside the female's tube if sperm swim in from the water, or it might happen externally after the female releases her eggs. The details vary among species, and for many tube worm species, reproductive biology remains poorly understood because it's so difficult to observe in the deep sea. Once fertilized, the eggs develop into larvae. These larvae are tiny, microscopic, and they're very different from adult tube worms. They're free-swimming, equipped with cilia, tiny hair-like structures that beat in coordinated waves to propel the larva through the water. The larvae don't have tubes yet, don't have plumes, don't have the adult body structure. They're trochophore larvae, a larval type common in annelids and some other groups, and they're built for dispersal, not for feeding or long-term survival. The larvae spend some time in the water column, drifting with currents, moving up away from the seafloor, or staying deep depending on the species. During this larval phase, they're vulnerable to predation, to being swept away to areas where no vents exist, to running out of energy before they find a suitable place to settle. They're living on yolk reserves from the egg, and those reserves won't last forever. How long the larvae remain in the water is not entirely clear. Estimates range from weeks to months. During this time, they can be carried great distances by deep-sea currents. The currents in the abyss are slow compared to surface currents, but over weeks or months, a larva could be transported tens or even hundreds of miles. This is how tube worms colonize new vents. When a fresh vent begins flowing, it's initially sterile, just hot water and bare rock. But somehow, within a few years, tube worms appear. The larvae must be drifting through the deep sea at low densities, and a few lucky ones happen to encounter the new vent. They detect the chemicals in the water, perhaps by chemoreceptors on their surfaces, and they respond by settling. Once settled, a larva undergoes metamorphosis. Its body reorganizes. It develops the structures needed for adult life. The trophosome, the circulatory system, the ability to secrete tube material, and crucially, it acquires its bacterial symbionts from the environment. Free-living bacteria, relatives of the symbionts, are present in vent waters, and the settling larvae take them in, establishing the partnership that will sustain them for the rest of their lives. Recent research has revealed something surprising about tube worm larvae. For a long time, scientists assumed the larvae dispersed through the open water far above the seafloor. But observations have found adult tube worms in unexpected places, specifically in sub-seafloor cavities beneath lava flows. These tube worms are living in spaces between sheets of cooled lava, fed by vent fluids percolating through the volcanic rock. The only way these worms could have gotten there is if their larvae traveled through the sub-sea floor itself, swimming or being carried by vent fluids through cracks and pores in the rock. This suggests a hidden dispersal mechanism, a subsurface network through which larvae might travel, potentially protected from predators and from being swept away by ocean currents. It also suggests that the vent ecosystems extend deeper into the crust than visible on the seafloor surface, that there are communities living in the rock itself, fed by fluids moving through the subsea floor circulation system. The challenges of larval dispersal mean that many larvae die before finding a vent. The deep sea is vast and vents are small and widely scattered. The odds of any individual larva encountering a vent are low, but tube worms compensate by producing huge numbers of offspring. A large female might release hundreds of thousands or even millions of eggs in her lifetime. Most of the larvae that hatch from those eggs will perish, but a few, a tiny fraction, will find vents and settle and survive to adulthood. This reproductive strategy, producing vast numbers of offspring with low individual survival rates, is common in marine invertebrates. It's sometimes called the lottery model of reproduction. You buy lots of tickets, the larvae, and you hope a few win by finding suitable habitat. The strategy works well in patchy environments where suitable habitat is rare but where successful individuals can thrive once they find it. For cold seep tube worms, reproduction is complicated by their extreme longevity. A worm that lives for 200 years is presumably reproducing for much of that time, contributing offspring to the population year after year. This creates overlapping generations, with newly settled juveniles growing alongside ancient adults, all participating in the reproductive cycle. It also means that genetic diversity can be maintained even in small populations, because the long reproductive lifespan reduces the effects of genetic drift. Understanding tube worm reproduction is important for conservation. As human activities move into deeper waters, as mining operations begin targeting hydrothermal vent minerals, as oil drilling expands into areas with cold seeps, these ecosystems face new threats. Knowing how vent and seep communities recover from disturbance, how quickly larvae can recolonize damaged sites, how far larvae can disperse, all of this information is crucial for managing and protecting these unique environments. And there's still so much we don't know. How do larvae detect new vents? What cues trigger settlement? How many larvae does it take to establish a new population? How connected are vent and seep populations across ocean basins? These questions drive ongoing research, with scientists using genetics, larval tracking, and sophisticated ocean models to try to understand the hidden lives of tube worms and other vent organisms. Living at a hydrothermal vent or cold seep requires dealing with conditions that would kill most organisms almost instantly. The combination of extreme pressure, temperature gradients, toxic chemicals, and complete darkness creates an environment that's hostile in multiple ways simultaneously. Tube worms have evolved remarkable adaptations to cope with these challenges, adaptations that make them true extremophiles among animals. Let's start with pressure. At the depths where most vents and seeps occur, typically between 500 and 10,000 feet below the surface, the pressure is immense. At 8,000 feet, the depth of the first discovered vents, the pressure is about 250 times atmospheric pressure at sea level. That's roughly 3,700 pounds pressing on every square inch. This pressure is enough to crush a sealed container, to compress gases, to affect the structure of proteins and cell membranes. Most deep-sea animals, including tube worms, deal with pressure through a simple strategy. They don't resist it. Their bodies don't contain gas-filled spaces that would be crushed by pressure. Their cell membranes and proteins are built to function under high pressure, with modifications to their lipid composition and protein structure that keep everything working normally despite the squeeze. In a sense, high pressure is their normal, and it's low pressure that would be problematic. A tube worm brought rapidly to the surface would experience dangerous expansion of any dissolved gases in its tissues, similar to decompression sickness in human divers. Temperature presents different challenges. At hydrothermal vents, the range is extreme. The water at the very mouth of a black smoker can be superheated to several hundred degrees. Just a few inches away, mixed with cold seawater, it might be lukewarm. A few feet away, it's near freezing. This creates steep gradients, with temperature changing dramatically over short distances. Tube worms position themselves in the zone where temperatures are tolerable, but where they can still access vent fluids. Their plumes extend into water that might be cool, while the base of their tubes might experience warmer water seeping up through cracks in the rock. The worms can tolerate a range of temperatures, but they avoid the extremes, staying away from the hottest vents while remaining close enough to benefit from the chemistry. Some vent animals, like the Pompeii worms, have special adaptations for heat tolerance. Their proteins remain stable at higher temperatures than typical proteins. They have heat shock proteins that help refold damaged proteins after heat stress. Tube worms have some of these adaptations as well, though not to the same extreme degree, allowing them to handle the warmth near vents without suffering damage. But the real challenge is chemical: hydrogen sulfide. The chemical that fuels the symbiotic bacteria is intensely toxic to most life. It binds to proteins in cells, particularly to proteins in the electron transport chain where cellular respiration happens. When hydrogen sulfide binds to these proteins, it shuts down the cell's ability to produce ATP, essentially suffocating the cell chemically even in the presence of oxygen. For most animals, exposure to hydrogen sulfide at the concentrations found in vent fluids would be rapidly fatal. Tube worms have evolved multiple mechanisms to handle hydrogen sulfide. First, their specialized hemoglobin, as we've discussed, can bind hydrogen sulfide without being poisoned by it, keeping the chemical safely contained during transport to the bacteria. Second, the bacteria themselves remove hydrogen sulfide from the worm's system by metabolizing it, preventing it from accumulating to dangerous levels in the worm's tissues. Third, the worms have enzymatic systems that can detoxify hydrogen sulfide, breaking it down or converting it to less harmful forms if concentrations get too high. The trophosome is essentially a contained chemical reactor, where hydrogen sulfide and oxygen, two substances that react violently together, are carefully managed and delivered to bacteria that can use them productively. The worm has to maintain precise control, providing enough hydrogen sulfide and oxygen for bacterial productivity without allowing toxic accumulation and without letting the two chemicals react prematurely outside the bacteria. Other vent chemicals are toxic as well. Heavy metals like copper, zinc, iron, and manganese are present at high concentrations in vent fluids. The low pH of some vent waters can damage tissues. There are various other reduced compounds, sulfides and sulfates, and things that form from them, all of which can be harmful at high levels. Vent animals generally have detoxification systems, proteins that can bind metals and sequester them safely, enzymes that can metabolize or neutralize various toxins. They also have robust cellular repair mechanisms, systems to fix damage to DNA, proteins, and membranes that might be caused by chemical stress or by the reactive oxygen species that form when oxygen-rich seawater mixes with reduced vent fluids. The darkness of the deep sea is perhaps the easiest challenge to adapt to. Many vent animals have reduced eyes or no eyes at all. Vision is useless without light. and the energy and resources needed to maintain eyes and a visual system are better spent elsewhere. Some animals retain simple light-sensing organs that can detect the faint glow of thermal radiation from hot vents, but most simply navigate by chemical sensing and touch, using chemoreceptors to detect gradients of vent chemicals and mechanoreceptors to sense their surroundings. Tube worms have no eyes, no need for them. They're sessile, stationary, incapable of moving to respond to visual information, even if they had it. Their awareness of their environment is purely chemical and tactile, sensing the water flowing past their plume, detecting changes in chemistry and temperature, responding by retracting into their tubes if conditions become threatening. The combination of all these stresses, pressure, temperature variation, chemical toxicity, darkness, creates an environment where survival requires constant physiological management. The tube worms' bodies are in many ways shaped more by chemistry than by physical structure, with their metabolism, their blood proteins, their symbiotic bacteria, all evolved to deal with a world where the normal rules don't apply, where what's toxic elsewhere is fuel, where what's impossible elsewhere is routine. The discovery of tube worms and their chemosynthetic ecosystems didn't just change marine biology, it changed astrobiology, the study of life in the universe. Before 1977, when scientists imagined where life might exist beyond Earth, they focused on places with sunlight, places with liquid water in the right temperature range, places that resembled Earth's surface. But tube worms showed that life could thrive without sunlight, could build entire ecosystems on chemistry alone, could survive in conditions previously thought uninhabitable. This opened up new possibilities. If life can exist at hydrothermal vents on Earth, powered by geothermal energy and chemical reactions rather than photosynthesis, then perhaps life could exist in similar environments elsewhere in the solar system or beyond. And there are candidates, places where conditions might mirror those at Earth's deep sea vents. Consider Europa, one of Jupiter's large moons. Europa is covered in ice, but beneath that ice is almost certainly a global ocean, kept liquid by tidal heating from Jupiter's gravity. The same tidal forces that flex Europa's interior and generate heat might also create hydrothermal vents on Europa's seafloor. If so, those vents could support chemosynthetic ecosystems, perhaps including organisms analogous to tube worms, living in the darkness beneath miles of ice. Enceladus, a moon of Saturn, is another possibility. Enceladus has geysers that erupt from cracks in its icy surface, shooting plumes of water vapor and ice particles into space. The composition of these plumes suggests they originate from a subsurface ocean, and the presence of certain molecules, including organic compounds and hydrogen gas, suggests chemical reactions similar to those at Earth's hydrothermal vents might be occurring. If Enceladus has vents, it could have life, simple microbes perhaps, but life nonetheless. Even Mars, cold and dry today, might have had hydrothermal vents in its ancient past when the planet had liquid water on its surface and active volcanism underground. If life arose on early Mars, it might have done so at vents, and perhaps microbes descended from those early organisms persist in subsurface environments today, living in groundwater, heated by residual geological activity, using chemosynthesis to survive. The idea of chemosynthetic life beyond Earth is attractive because it doesn't require the complicated conditions needed for photosynthesis. Photosynthesis requires a transparent atmosphere, a stable source of bright light, and the evolution of complex molecular machinery to capture that light and convert it to chemical energy. Chemosynthesis, by contrast, requires only geological activity, water, and the right mix of chemicals. These conditions might be common in the cosmos, far more common than the conditions needed for photosynthetic ecosystems. Tube worms also inform our thinking about the origin of life on Earth. Many scientists now believe that life might have originated at hydrothermal vents or in similar geothermal environments. The argument goes like this. Early Earth was a harsh place with intense UV radiation from the sun, no protective ozone layer, and a thin, possibly unstable atmosphere. But deep underwater at vents, conditions would have been more stable, protected from radiation, with abundant chemical energy and minerals that could catalyze organic reactions. The chemical gradients at vents, the mixing of reducing fluids with oxidized seawater, create natural energy sources similar to those used by chemosynthetic bacteria. Some scientists propose that the first self-replicating molecules, the precursors to life, might have formed at vents, using minerals on chimney surfaces as scaffolds, using the energy from chemical gradients to drive synthesis of complex organic molecules. If this hypothesis is correct, then chemosynthesis is not just an alternative to photosynthesis. It's the original metabolism, the way life first powered itself before photosynthesis evolved. And tube worms, living at vents today, might be distant descendants of the earliest living things, carrying on a lifestyle that dates back billions of years to the dawn of life. This perspective makes tube worms more than just interesting animals. They're windows into deep time, into the early history of life on our planet. Studying them, understanding how they and their bacterial symbionts make a living, helps us understand what early life might have been like, what challenges it faced, what solutions it evolved. And if we ever discover life elsewhere in the solar system or beyond, if we find microbes in Europa's ocean or fossils in Martian rocks, and if that life turns out to be chemosynthetic, living on chemical energy in the absence of light, we'll know that such life is possible because we've seen it on Earth. Tube worms and their vent communities will have provided the proof of concept, the existence proof that life can thrive in such environments. They expand our sense of where life might be found, the range of conditions that can support living things. They remind us that Earth itself still holds mysteries, that there are frontiers of biological discovery in our own planet's oceans, and they inspire hope in a way because they show that life is resilient and creative, that evolution finds ways to survive and thrive even in places that seem impossible. In the deep sea darkness, far from the sun, tube worms stand as proof that life doesn't need light, doesn't need gentle conditions, doesn't need anything but chemistry, water, and time. They are testaments to life's stubbornness, its refusal to be limited by our assumptions about what's possible, and they are, in their own quiet way, among the most remarkable organisms on Earth. We've traveled far tonight, from the sunlit surface, down through miles of water into the permanent darkness of the abyss. We've met tube worms, creatures without mouths, without stomachs, without eyes, creatures that survive on chemistry and symbiosis in one of the most extreme environments on the planet. We've learned about hydrothermal vents where superheated water rich in toxic chemicals creates oases of life. We've discovered cold seeps where tube worms live for centuries, quietly filtering water and housing their bacterial gardens. We've seen how life, stubborn and inventive, has found ways to thrive without sunlight, how chemosynthesis can power entire ecosystems, how partnerships between animals and microbes can create possibilities that neither could achieve alone. We've glimpsed communities of clams and crabs and shrimp, all adapted to the strange conditions of vents and seeps. We've followed the journeys of lava, drifting through the deep ocean in search of new homes, and we've considered what these discoveries mean for our understanding of life in the universe. How tube worms and their ecosystems expand the range of places where we might search for life, how they inform our thinking about life's origins on our own planet. But perhaps most importantly, we've been reminded of how little we know, how much remains to be discovered even in our own world. The deep ocean covers most of Earth's surface, yet we've explored less than 20% of it in any detail. There are vents still waiting to be found, seeps not yet mapped, species not yet described. Every expedition to the deep brings back surprises, creatures never before seen, behaviors never before observed, ecosystems whose existence challenges our assumptions. The tube worms are part of this hidden world, standing in the darkness, anchored to rock, their plumes waving gently in currents we will probably never feel, living lives that span centuries, quietly performing their role in cycling chemicals between the Earth's interior and the ocean, connecting the geology of the planet with the biology of the sea. They ask nothing from us, these worms. They don't require our attention or understanding to thrive. They were living at vents and seeps for millions of years before humans existed, and they'll likely continue long after we're gone, as long as Earth remains geologically active, as long as vents keep flowing and seeps keep seeping. But we can learn from them, we can be inspired by them, we can let them remind us that life is more varied, more resilient, more creative than we often give it credit for. We can let them expand our sense of what's possible, where life can exist, what it can do. Tonight, as you drift towards sleep, picture those tube worms if you like. Imagine them in their colonies at the bottom of the sea, swaying in the current, their red plumes bright against the white tubes, against the black volcanic rock, against the darkness that surrounds them. Imagine the bacteria inside them, billions of microscopic partners working invisibly to convert poison into food. Imagine the vents, the plumes of shimmering water, the strange creatures that call these places home. And rest. Let your breathing slow. Let your thoughts settle like sediment drifting down through water. Let yourself sink into comfort and warmth, because the ocean's mysteries will still be there tomorrow, waiting. The tube worms will still be standing in the abyss, doing what they've done for eons, living without light, thriving without sun, proving that life finds a way even in the deepest darkness. Sleep well, dream of strange and hidden wonders, and know that the world is full of miracles, some of them so quiet and so far from sight that we only discovered them yesterday, and yet they've been there all along, in the dark, in the deep, waiting patiently for us to arrive and bear witness to their existence. The ocean keeps its secrets well, but sometimes, if we're lucky, if we're curious, if we send our submarines down into the darkness, we get to glimpse them. We get to see tube worms and black smokers and communities powered by chemistry. We get to learn that life is stranger and more wonderful than we imagined. And that's a good thought to fall asleep to. That the world is full of marvels, that mysteries remain, that there are still discoveries to be made, still wonders waiting in the depths. Good night.